Hello, everyone. Good evening. Good evening to everybody. Am I audible, everyone? Good evening, everybody. Can I have some quick responses? Hello, Ashwarya. Good evening. Welcome to the session. Good evening, all the students. If you can hear me, please give me a quick response. Yeah, good evening, Anusha. Good to see you here. Good evening, Surendra. Good evening, Saujanya. Okay, I think people are still joining. Let them join. So, today we are going to discuss some mixed bag questions from Unit 11. So, today we are not going to separate the questions on the basis of their subunits because we have kind of finished Unit 11 completely. So, we will see some mixed bag questions. Okay, it might be from any subunit, it might be a combination of topics that we have covered in multiple subunits, but it is from Unit 11. Okay, so those kind of questions we will see. So this will be like a revision, and let me tell you that all of these questions, every single question that we are going to discuss today, are all CSIR paper questions. Okay, and most of them are from the recent CSIR paper. Okay, last 2 or 3 years paper only I have taken questions from. Okay, so these are all the questions, most all the questions from Unit 11, like the difficult ones specifically I have chosen, so that I can tell you, I can show you how to solve. Okay, so one thing that is that will be common in today's questions, you will see that it will be from any topic, any subunit from Unit 11. Apart from that, you will also notice that all these questions, because in Unit 11 usually the questions can be numerical based, questions can be statement based, and one more thing that is usually asked is graphical questions or graph based questions. Okay, so where you are talking about any kind, it can be any phenomena, whether it might be something related to speciation, it might be something related to a natural selection, it might be something related to parental care, it might be something related to sexual selection. Okay, it can be anything, but the information is given to you in words and then some graphical representation is there. Okay, so in today's session we are specifically going to focus on those kind of questions which are going to which involves a graph. Okay, so you should have a very good idea about how to read a graph and how do we take out the information from that. Okay, so that knowledge you will not only apply in this kind, like in today's session or only in Unit 11 questions, you will get to apply in a lot many questions that come in CSIR papers, which are analytical questions. Okay, so that basically checks how good you are in handling data, how good you are in collect in taking out information from the graph. Okay, and that is actually how most of the scientific studies they represent their result, right? So this is very important for you to understand the graph and also to represent your findings in the form of graph. So both the analysis, so graphical analysis is for sure a very important part, and in today's session we are going to mainly focus on that. So I see some more people. Yeah, good evening, Dipshika, Shivani, Sunshine, Madhugula. Good evening everyone. Okay, so are you all ready? So as always, I want your active participation also. So definitely I will tell you how to solve the question and what should be the answer. But before that, I want your active participation and I want you all to answer as well. Okay. And it's absolutely okay if it is wrong, because when you do mistakes, that is when you learn. Okay. But you have to learn today. All right. Starting. Let's start our session. So first question. So here there are two set of graphs given. Okay, I'm sorry, the graphs are not very clear because I have actually taken it out from the question paper. So it is little blur, but I hope you will be able to read the headings here. So the top panel, that is from graph A to C. This one, okay, this is the top panel. Represent the trends of number of sperms produced per mating season with respect to number of mates, while the bottom panel, okay, this is the bottom panel from 1 to 4, it represents trends of time invested in parental care with respect to number of mates in birds. Okay, so select the trend from each panel. Select the correct trend from each panel. Okay, so in case of birds, what is the trend we see in terms of number of sperms produced compare versus number of mates, and time invested in parental care versus number of mates. Okay, so what is the relationship that we see in the birds population? That is what we have to choose, or which graph actually. Okay, from top panel one graph, from bottom panel one graph, which is representing the correct trend that we see in the birds population. That is what we have to choose. Yes, anyone? Excellent. Achal, Surendra, Anusha, Sunshine. Yes, Dipshika. Yes, all of you are correct. Okay. Now, what is the comparison that we know? So, definitely, when the number of mates will increase, the number of sperms produced per mating season will be high, right? So more is the number of mates the male is mating with. Okay, so definitely the number of sperms produced by the male will also be high. Okay, so there is a direct correlation between number of sperms is directly proportional to number of mates. Okay, so what does that mean? More is the number of mate, more will be the amount of sperm produced. Okay, now in this top panel graph, if we see, only graph A is the one which is showing the direct proportional relationship between the number of sperms and number of mates. Okay, in this case, what do we see? That initially it decreases, okay, and then it increases, but this decrease, it will never happen. This will not happen. Okay, and in the third one, we see that there is an increase initially, but then it decreases drastically. That is also something which will not happen. Okay, so this will be a direct correlation. Okay, next parameter what we have, time invested in parental care. Okay, so time in parental care. Now, what will be the relationship here? See, the thing is, in parental care, what the individual is doing, it is taking care of the young one. So definitely, if the individual is taking care, is occupied in taking care of the young one, so definitely he or she will not have time for courtship or to attract the other gender, or to mate. Okay, so more is the time invested in parental care, lesser will be the number of mates. Okay, so time in parental care is inversely proportional to number of mates. So definitely, if number of mates are more, okay, so if the individual is actively participating in mating, okay, with multiple partners, that means that person will not have any time to take care of the young ones. Okay, so there the time in parental care will be less. Okay, so number of sperms is directly proportional to the number of mates, but the time spent in parental care is inversely proportional to the number of mates. So if we see this graphs in the bottom panel, so which graph represents this finding? So this will be graph number 2. Okay, is it understood, everyone? Give me some quick thumbs ups, and that will be enough. That will make me know that you have understood. All right. Next question. Directional selection of a particular trait will lead to the frequency of the trait being normally distributed in the population, always showing skewed distribution in the population, always showing right skewed distribution in the population, showing either right or left skewed distribution in the population. Here it is asking about directional selection. Now, this was a question which was asked in the June 2023 exam itself. Yes. So, what is this directional selection? So, directional selection means that whatever is the average phenotype of this generation. So, after one or two generation of directional selection, the average phenotype of the population will change. Okay. Now, see here, for example, it is showing the beetle color. Okay. So, this is the spectrum. Okay, from light green color to dark green color. Okay, so suppose in generation 0, okay, so this was this phenotype. Okay, this shade of green was the phenotype that was shown by the average population. Okay, maximum individuals of the population. But after directional selection, what happened? So maybe this phenotype was having some selection disadvantage, and because of that, now after some generation, so this is the after selection. Okay, this is after selection. So after selection, suppose this phenotype, okay, whatever color of green it is, so definitely this color of this shade of green is darker than this. Okay, so this dark shade of green became the average phenotype. Okay, so this is directional selection where the average phenotype changes. Okay, but this is one example shown here. Now, if in a different population, okay, if this happens, okay, where the average population, the average phenotype of the population became lighter in color, okay, that will also be called as directional selection, right? What is important here, that the average phenotype will change, okay, to a different average phenotype. Okay, only one average value. Okay, so from the average phenotype, it can change in either of the direction. That will be called as directional selection. Okay, but only one direction it will move, not both, because if it goes in both, then we call it as disruptive selection. Remember, where the extreme phenotypes are selected and the average phenotype is not selected. Okay, but since in case of disruptive selection, that would have been in both direction, it will move in both direction. But since it is directional selection, so it will be in either direction, either left or right. Okay, so what will be the answer will be that it will show either right or left skewed distribution in the population. Did you all understand? So in case of directional selection, if it was disruptive selection, so what will be your answer then? So if it was disruptive selection, then the answer will be in both direction, both right and left. Got it? Okay. Yes, so before going ahead, I am very sure you all are already knowing that the Watson Junoon scholarship is going on right now. So the enrollment of that is going on right now. So you can enroll, and if you people are already enrolled, because I see many of you are already our students, so please tell your friends and anybody you know who wants to pursue, who wants to take admission in Biotechnica for CSIR NET or GATE classes, or even the IIT JAM Common University Entrance Exam or GAT-B exams. Okay, and then along with that, you already might be knowing that you will get the access to scholarships, even R programming internship you will get, 4 certification courses you have got. Okay, and then you will have the 24/7 doubt solving sessions along with the VIP pass for our CRISPR conference. So any further information, and the scholarship is also there. Okay, you might get the scholarship, and for any more information you can call us in this toll free numbers for more information. Okay, but yes, the time is running, guys, so if you are really interested, please join it as soon as possible. And huge discounts are also going on. Okay, you will get a you will have to give a scholarship interview. On the basis of that, you will you will be given scholarships also. You have the opportunity to win scholarships also. Okay, so next question. Reduction in the frequency of heterozygous genotype with a concomitant increase in the frequency of homozygous genotype in context of random mating is due to. Reduction in the frequency of heterozygous genotype and increase in the frequency of homozygous genotype in context to random mating is due to. Yes, absolutely correct. The answer is intense inbreeding. Okay, so if it is not random mating, then what is the other option? It has to be non-random mating. Okay, or we can also call it as selective mating. So if it is non-random mating, okay, then slowly the heterozygosity will decrease and homozygosity will increase. Okay, but this decrease in heterozygosity and the related amount of increase in homozygosity will be drastic if there is inbreeding, which is the most extensive form of non-random mating. Next question. This is also a CSR question, Alin. So it's quite an interesting question. Uh, Inclusive fitness of an animal can be measured as a sum of direct fitness and indirect fitness. Imagine you have 10 offsprings. Through diligent parental care, 5 survive to reproduce. You give your life in a heroic deed to save a total of 5 of your nieces and nephews. What is your inclusive fitness? So, this question here is talking about altruism, which is a self-sacrificial behavior. Okay, a special type of altruism when it is done for blood relatives is called as kin selection. Okay, so this example where you are sacrificing your life to save your nieces and nephews. So nieces and nephews are the ones who with whom you share your genes. They are your blood relatives. Okay, so this is an example of kin selection, that you are doing your own sacrifice to save your kin. Okay, so what will be the inclusive fitness? So for inclusive fitness, it is given direct fitness and indirect fitness, right? So it is already told here that inclusive fitness is equal to direct fitness plus indirect fitness. Okay. Now, what will be direct fitness? So direct fitness you will get from your offsprings. Okay, your offsprings will give you direct fitness. Okay, so what will be the value of direct fitness? So I'm writing it here. Okay, so direct fitness will be from your offsprings. Okay, so the fitness, how do we get it? It will be the number of offsprings that you are that are surviving to reproduce, multiplied with the genetic relatedness between you and them. Okay, so direct fitness you are getting from your offsprings. Genetic relatedness between you and your offspring is 0.5. Okay, now you have 10 offsprings but only 5 survive to reproduce. Okay, so that number of offsprings that survive to reproduce, okay, that from them only you will get the fitness. Okay, the ones who you have reproduced but they are not alive to reproduce means they cannot pass on your genes. Okay, so they will not give you fitness. The fitness you will get from the offsprings who are surviving to reproduce. Okay, so direct fitness will be 0.5 multiplied by 5 because only 5 are surviving. Okay, do you understand? Sunshine, Sanket. So direct fitness you will get is 0.5, which is R. That is genetic relatedness between you and your offspring, multiplied with 5 because 5 are surviving till for reproduction. Okay, so it will be equal to 2.5. Now, indirect fitness you will get from your niece and nephew whom you have saved by doing your sacrifice. Okay, and the same way as in direct fitness, here also you will get it by multiplying genetic relatedness with the number of children or number of keens that you have saved. Okay, now nephew and niece, what is the value of R for that? Yes, that's correct, Anupam. What is the value of R for nephew and niece? It is 0.25. Okay, so 0.25 again multiplied by 5 because you saved 5 of them. All right, so it will be 1.25. So inclusive fitness will be 2.5, that because that is the direct fitness you are getting, plus indirect fitness 1.25, so that will be 3.75. Understood? So is it not a good question? That checks your understanding, as well as it's a numerical, but the definitely the multiplication or the math part is not at all difficult. What is the main key here is the concept and how you are applying it. Next question. Any movie that features dinosaurs should also have which of the following combination of geological age appropriate organisms? Correct, sorry, choose the correct combination. Okay, so just you have to imagine that you are watching Jurassic Park, and provided Jurassic Park directors, they have done the proper research and they have checked what all flora fauna was present at that time. Okay, so what all geological age appropriate organisms you will see. So this is a question from the paleontological history. So here you don't have to actually go and watch Jurassic Park to answer this question. Here basically you have to think about what all organisms were present at the time when dinosaurs were there. So dinosaurs are there means we are talking about the Mesozoic era. Okay. To be specific, we are talking about the Jurassic and the Cretaceous period. Okay, so now you have to think along with dinosaurs what all organisms were present during the Jurassic and Cretaceous period. Okay, so what all were there? Humans, angiosperm, gymnosperm, birds. For sure, no. Humans for sure were not there. Okay, apes, gymnosperm, birds. Again, apes were not there. Okay, uh, if it was written small mammals, okay, that could have been the case because mammals were present, but apes for sure not. Early diverging gymnosperms, amphibians and reptiles. Amphibians, reptiles were present, but early diverging gymnosperms, that happened way back in the Paleozoic era. So right now we are in Mesozoic era, so it is the time where the early diverging angiosperms were there, reptiles and amphibians. No, Anupam. So you have to understand that Jurassic Park does not say that it is in that era. It says that now by biotechnology we have created a park where dinosaurs are present. Okay, it does not say that you go back to those many million years ago when dinosaurs were dominant on Earth. It says that now in human world we have created an amusement park with real dinosaurs. Okay, moving on. Next question. So this question was also there in the last CSR exam. In four taxa A, B, C, D, two characters, shape and color, were scored to infer their phylogenetic relationship. The two character states for shape were square and round, while the two character states for color were black and yellow. So shape was square and round, color was black and yellow. Okay, the character distribution is given in the table below, right? So these are the different species MNOP. So what shape, what color they. Now using the above data, four trees were built using the method of maximum parsimony, which are given below. Okay, A, B, C, and D. Select the option that represents the two most parsimonious tree. Okay, so usually when this kind of question used to come, so they used to also mark the evolutionary event by in the form of these horizontal lines, right? So if that was there, then it is very easy, we can directly calculate the number of evolutionary events and from there we can calculate. Okay, but this time it went one step ahead where the table is given and you have to, from the table you have to actually first find out what all evolutionary events are occurring and where all it can take place in the phylogenetic tree. Okay, you have to first mark the evolutionary events and then you have to count and find out the most parsimonious tree. Yes, what do you think will be the answer? Yes, there's a principle of maximum parsimony, Ramdas, which says that the phylogenetic tree with the least number of evolutionary events is the most probable tree. That is the principle of maximum parsimony, which is usually used to find out the most probable tree. So in this question, okay, so first thing what we have to do, we have to mark the evolutionary events, right? So what do we see here? So first of all, we have to quickly check whether the trees are made correctly or not. Okay. So M is black and square. So M is black and square. N is yellow. So all the other trees are yellow. So M and O, yellow, yellow, yellow. In that only O is square. Other twos are circle. Okay. So we see that the trees are correct. At least the features that are mentioned for them are correct. Okay. Now what we have to do, we have to mark the evolutionary event. Okay, so if this is the tree, right, so that means M here. So for A, what it means that M is the outgroup. So whatever features M has, most probably the common ancestor also had it. So common ancestor was black and square. Okay, so here all other trees are yellow. That means here one evolutionary event happened where the color changed to yellow. So color became yellow. Sorry. Okay. Now what happened? So color became yellow. Okay. And then what happened? So the shape is still square. Okay. But here these two are circle, means they are common ancestor. In that also one more evolutionary event happened where shape became circle. Okay. So there are two evolutionary events in this tree A. Okay. Likewise, let's mark tree B. So in there, P is the outgroup, which is yellow and round. Okay. That means here one evolutionary event happened where the shape became square. Okay. For the shape, one evolutionary event happened. Okay. The color remained same. Now here, one evolutionary event happened which made it black. Okay. And one evolutionary event happened which made it circle. One more evolutionary event happened for shape. Okay, so B has three evolutionary events. Yes, Karen, Sunshine, Dipshika, Surender, yes, all of you are correct. Now for tree C, okay, let's see the evolutionary event likewise. So this is correct. So that means here one evolutionary event happened for the color. Okay, so where color became yellow. Then one more evolutionary event happened for sure where it became circle as well. Okay, so two evolutionary event happened here. So one was for shape, one was for color. Okay, and then you see one evolutionary event happened in O, only in the branch leading to O, where it again became square. Okay, so this also has three evolutionary events. For D, if we see, so you can see one evolutionary event happened where it became circle. Okay, and this remains square. And one evolutionary event, sorry, okay, so they all are yellow, means the common ancestor was also yellow. That means one derived uniquely derived characteristic for M was black. Okay, so two evolutionary event, right? So now once you have marked the evolutionary events, now you calculate. Okay, so for A we have two, for B we have three, for C we have three, for D we have two. Okay, so which are the two most parsimonious tree will be the ones with the least number of evolutionary events, meaning A and D. Option 4. Okay, so Anusha, actually we have solved this kind of questions before also in this YouTube session as well as in class as well. But in those questions, what was the thing that the evolutionary events were mentioned? Okay, so this you can say one step ahead, and it is one step more complicated, where you have to study the table and first mark the evolutionary events, and then you have to count for finding out the most parsimonious tree. Okay, next question. Character similarity that can be misinterpreted as common descent is called, this is quite easy. That can be misinterpreted. Okay, it is not common descent, but it is misinterpreted as common descent. What will the answer be? Symplisomorphy, synapomorphy, homology, and homoplasy. Now, what do you mean by common descent? Common descent means coming or inherited from common ancestor. Okay, so synplisomorphy, what is it? It is shared ancestral character. Okay, it is already having common descent. Synapomorphy is shared derived character. Okay, so since it is shared derived character, so that also is coming from the common ancestor, but it is not the true ancestor. It is from the most recent common ancestor. Okay, so this is also having common descent, this is also having common descent. Homology also has common descent, right? Because homology means what? That the structure have similar structure, the characteristics which have similar structure but different function. So since they have similar structure, that means they shows common descent. So all this have common descent. But what about homoplasy? Homoplasy happens when two unrelated groups or two very distantly related groups, they start showing the same character. Okay, that is what is called as homoplasy. So you'll remember, usually we will represent homoplasy like this. Okay, so same character came in two different species which are distantly related. Okay, and none of them got it from their common ancestor, right? So that is an example of homoplasy. So homoplasy is a character which can be misinterpreted as common descent. Okay, all other terms that are given, they are already common descent only. There is no misinterpretation there. Next question. So as I told you, we'll be talking about a lot more graphical questions. So this is also a graphical question here. A particular behavioral variant affects fitness of an organism. The relationship between the frequency of the variant in the population and fitness are plotted below. So what it is, so whenever there's a graphical question, so it is very important that you understand what are there in the coordinates. Okay, because that will only give you the information. So it is very important. So if you read the question and you do not understand the coordinates, then stop, read again. Okay, it's very important. The first thing to understand the graph is to understand the coordinates. So what are the coordinates we have? So the graph is plotted between the frequency of the variant in the population and the fitness. Okay, so in which of these cases is the behavioral variant most likely to reach a frequency of 1? Okay, now you guys tell me, what do you mean by frequency of 1? What is the meaning of this? Behavioral variant having a frequency of 1. What do you mean by that? No, Anupam, first I asked you, tell me what do you mean by frequency of 1? Frequency of 1 means staying alive. No. Absolutely correct, Suzanne. So when I say frequency of 1 means that is the only variant which is present. Okay, basically that will mean that 100% population has that particular variation. It is present in 100% of the population, right? That is what we mean by frequency of 1. Okay, so when it is reaching the frequency of 1, okay, that means its fitness is also increasing, right? So when it is getting continuously distributed in the population, so its fitness is also increasing. That is when it is possible that 100% of the population has it, right? So when we see the relationship between fitness and frequency, what we will see that as the frequency increases, so in this case, frequency will be directly proportional to fitness, right? So as the frequency increases, the fitness also increases. Okay, and that is when they can achieve a 100 frequency of 1, that is the entire population showing that particular phenotype. Okay, so when which of the graph is showing the this direct proportional relationship is only graph C, where fitness also is increasing and the frequency is also increasing. All right, moving on. I really hope that you all are getting it. Yes, everyone, are you understanding all those questions which we are discussing? Okay, great. Yeah. So next question we are going to talk about is the matching between the columns. Okay. So match the geographical time period with the extinction or diversification events associated with them. Okay. Again, this is from the paleontological history. So Cenozoic era, Cretaceous period, Paleozoic era, and Quaternary period are mentioned. Okay. Now let's see the events. Angiosperm diversification. So angiosperm showing their first appearance that happened during the Cretaceous period, but angiosperm diversification that happened during the next era. Okay, so when we are basically talking about the Cretaceous, sorry, the angiosperm diversification. Okay, so that will happen during the Cretaceous period only. Okay, so that is the time when the when it showed the extensive diversification. Okay, so it started, the diversification of angiosperm started during the Cretaceous period. Okay, now next what we have, modern fauna diversification. Okay, bivalves, gastropod, bryozoan. Malacostracan, crustaceans. Okay, so this modern fauna diversification happened during the Cambrian explosion itself. Okay, so during the Paleozoic era. Right? Now, next, megafauna extinction. So, megafauna extinction, yes, here basically we are talking about a mass extinction. So, the major mass extinction for sure happened during the Permian period, but at the end of the Permian period, but that we don't see in the option. Okay, so apart from that, one fauna extinction that we can see is happening is right now. Okay, during the Quaternary period. And fourth is mammalian diversification. So that happened during the Cenozoic era. Okay, so what it is: A4, B1, C2, D3. Answer is option B. So I see only Karen has given the correct answer. What about others? Okay, so are you people practicing the paleontological history? Are you following the tip that I asked you to do, that take an image and put it up in in the room where you can see it and revise it for 5 minutes every day? Anyone who is following that? Yeah, I know you have to revise, because here it is not much of a concept or anything more conceptual that you have to remember. It is more of remembering the things, okay, just to names and everything. So this is less history, okay, and in history you have to remember the dates and all. So here you don't have to remember the dates, but you have to remember the events. Okay, so that is why I told you that that image that I showed you, or which is there in the PPT. So just take a printout of that, put it up there in your room somewhere, where you can see it, and revise every day for 5 minutes. Okay, next question. So this is also a graph based question, as you can see, and you will see that when you can read the graph, there cannot be any question which is easier than this. So given below are graphs depicting two possible dynamics of gene duplication events over a period of time during genome evolution. Based on the above figures, which one of the following options correctly represents the identity of A, B, C, and D? Okay, so A, B, C, and D, these are some times which are mentioned in the graph. So first, what we will do, check the graph and see what is there in the coordinate. Okay, so in the x-axis, we have time since duplication event, and on the y-axis, we have number of pairs of duplicated genes. Okay, so what do we see here? That immediately after, so basically the graph will, the x-axis will grow like this, right? So in this direction, the time since duplication is increasing. Okay, so that means this is the time, okay, where duplication event has actually happened. So this is the time where duplication happened. Okay, so what we have, right? So A, you can see that the number of pairs of duplicated gene is very high. Okay, because this is y-axis, it will grow in this direction. So here it is very high. Okay, so immediately after duplication, the number of duplicated genes are very high. Okay, so A will show a gene duplication event, right? Now, what is B showing? B is the time where the number of duplicated genes are depleting. Okay, so why it is happening? Because of loss of duplicated gene can be one of the answer. So here the duplicated genes are getting lost. And C is the what? So after the losing of the duplicated genes, what all duplicated genes remained, that is shown by C, right? So C is the remaining pair of duplicated genes. Now in a different graph, there is D also. So every time there is D, so there are two Ds. Okay, so in that case, what is happening? There is a peak in the graph, and that peak is increasing in the number of duplicated genes here. Okay, that means D are additional duplication events, or it is an additional duplication event. Okay, so what is A? The gene duplication event. B, loss of duplicated genes. C, the remaining pairs of duplicated genes. And D, additional duplication events. Okay, so answer will be option A. So you can see in this question, right, you don't have to have any information. You don't have to know the evolution of duplicated genes. You don't have to talk about orthologous, paralogous, nothing. Okay, just by simple logic and understanding of the graph, you can answer. Okay, next question. Given below are proposed analogous structures among organisms. Which one of the following option correctly states the analogous structures? Okay, so basically there are 5 comparisons are given. Okay, A, B, C, D, E, and from there you have to identify the pair of analogous structures. Okay, so for that, again, you have to have an idea what are analogous structures. So analogous structures are those structures having different structure but same function. Okay, so basically in the unrelated organisms or distantly related organisms, if the similar feature comes back, then it is called as analogous structure. Okay, so that actually happens when individuals of different species, they live in the same ecosystem. So wings of bat and bird, bat and tetrapod, digits, tendrils of whitish and tendrils of pumpkin, tubers of potato and sweet potato, fins of fish and flippers of whale. What all will be the analogous structures? So the first thing that you will know is that bird and bat, okay, they are analogous. So they both are used for flying, but the anatomical structure of birds, okay, that we will see, the wings of bird is very much different from the anatomical structure of the wings of bat. Okay, birds will have feather and all, and also the bone arrangement is different. Okay, so wings of bird and bat is for sure one analogous character. Okay. Next, wings of bats and tetrapod digits. So wings of bat and tetrapod digits, okay, when we see, they are homologous characters, right? Because bats are mammals, and all the mammals, okay, when we see the four limb of the mammals have the same anatomical structure. Okay, so B is an example of homologous character. Next, tendril of whitish and tendril of pumpkin. Okay, so these tendrils are what you might have seen that from the stem, this kind of things will come out, right? So they are called as tendrils. Okay, now when we see the tendril of whitish, which is from the group whitish, like the grapevine. Okay, so when you see the tendril of grapevine and the tendrils of pumpkin, so they both come out from the same portion of the stem. Okay, so they have similar structure, but their function is different. So tendril of whitish is used for support, and tendril of pumpkin is used for climbing. Okay, so they have different function. So tendril of whitish and tendril of pumpkin are also homologous character because they have same structure but different function. Hi Naresh, welcome to the session, but I will say that you joined little late. Okay, anyways, we are still discussing questions and you can take the advantage of that. So moving on, the D is tubers of potato and sweet potato. So potato and sweet potato, they are both tubers, but they are different. Potato is the modified stem, whereas sweet potato is modified root. Okay, so sweet potato is modified root, but potato is modified stem. Okay, so different function. Then so this A is analogous, D is analogous. So they have similar stress function. Sorry, they have similar function that they both store food and nutrients for the plant. But in potato it is modified stem, but in sweet potato it is modified root, different structure. Okay. Now E, fins of fish and flippers of whale. So again, you here also you can see the structure of fin and whale. The fish fin and whale fin. So they are structure anatomically different, but both of them are used for swimming. Okay, so in fact the whale's fin, if you compare with the wing of bat, then it will become homologous because their anatomical structure is similar. Okay, so A, D and E are analogous structures. Okay, moving on. Next question. Assume a male sparrow X is hatched and reared in isolation and allowed a critical imprinting period to hear the song of male of another sparrow that from species Y. Now, after the isolation, what kind of behavior will species X show? Okay. So what do we see here? That in case of birds, okay, not only for sparrow, in all the birds, they have for them this singing is very important. Okay, so what is this singing? So this singing is definitely not for our entertainment. It is for attracting the mates. Okay, and it is also for communication between the group, right? So this so this song is a type of it's the courtship or behavior that is done by the birds, right? So there is a male sparrow which is hatched and reared in isolation and allowed a critical imprinting period. Imprinting period means what? Immediately after hatching, the newborns have an imprinting period. Within that period, they identify their parents. Okay, they learn to differentiate their parents from other adults of the species. Okay, so this imprinting period is important. So during the imprinting period, this male sparrow X listens to a sparrow of a different species. Okay, now all the species, they have their species specific song. Okay, right? So what will be the song that this species will show now? Okay, so in this case you have to understand that yes, imprinting period is important. Okay, but this imprinting period is used to kind of fine tune the skill that is innate, which is already present in the sparrow, in the birds. Okay, even the newborn birds. Okay, because this ability to sing is something which will be genetic. Okay, which song to sing will also be genetic, right? But the tune of that will not be that much that much clear or that much fine tuned. Okay, so in the sparrows, the newborn sparrows, they will listen to the adults of that same species and they will fine tune their songs. Okay, so this genetic element is present in the male sparrow even when it is reared in isolation. Okay, now during the fine tuning period, during the imprinting period, it is listening to song Y. Okay, so what will happen? So it has been seen in the studies that this species X will now sing its song only in the tune of Y. Okay, so what are the options given? It will sing us the song of species Y that it has heard in the critical period. No. It will sing the song of its own species X only. No. It will not sing at all. No. It will sing a song which is not sung by either X or Y. Okay, because either X will sing what? It will sing the song X in tune X. Y will sing the song Y in tune Y. Okay, but in this condition, when a X sparrow is reared in isolation and it listens to the song of Y during imprinting, so it will sing the song of X in Y's tune. Okay, so it will be a new song which is not sung by either X and Y. Okay, so answer will be D. Right. So let's yes, this is a very good question. Let's see this question before we end the class. So, a founder population has an heterozygous genotype with a frequency of 1 and no individual with either homozygous dominant or homozygous recessive genotype. With repeated self fertilization, the frequency of all the three genotypes after three generation, what it will be. Okay, so when we are talking about founders population, means we are talking about founders effect. So in case of founder effect, which evolutionary force is most active? Genetic drift, right? Now in case of genetic drift, what is the way, or in which proportion the heterozygosity is lost? So heterozygosity is lost by a value of 1 by 2 every generation. Okay, so now we can make a table. Okay, so this is heterozygote, this is homozygous dominant, this is homozygous recessive. Okay, so during generation 0, so this is 1, so this 2 were 0. Okay, now during generation so after first generation of genetic drift, what will happen? This will become half. Okay, so this became half, so half of that is added, so half of half is 1 by 4. Okay, so it is basically 0 plus 1 by 4 will be added, will be the frequency of homozygous dominant. Similarly, 0 plus 1 by 4 will be the frequency of heterozygote, sorry, homozygous recessive. Okay, now for generation 2, what will happen? So this value will again become half, so half of half is 1 by 4. Okay, and again it will be added, whatever is reduced in heterozygote, that will be added in homozygotes. Okay, but there are two homozygotes, so in each homozygote half of that will be added. Okay, so it will be again 1 by 4 was already there from generation 1, so 1 by 4 plus 1 by 8, half of 1 by 4. Okay, so like that, when you keep on doing for the third generation also, so heterozygote frequency will become 1 by 8, and homozygous dominant and homozygous recessive frequency will become 7 by 16 respectively. Okay, so this is how you have to find out, right? Okay, so thank you everyone. We come to the end of the session here. Okay, from so I have tried to take up questions, all CSIR based questions from Unit 11. Okay, so I hope all these sessions you are finding them helpful and you will keep joining them. And these sessions are completely free. So tell your all other friends also to join if they want. Okay, and this is the information about our Watson Junoon Scholarship. Thank you everyone once again for your time and for attending the session. Bye bye. See you in the next class.